This is an OCR A-level biology video on proteins today. We're also going to be looking at inorganic ions. So proteins are formed from amino acids, which are the monomers of all proteins, and all amino acids have the same basic structure, which I'll speak to you about in a second. So a bunch of amino acids join together to form polypeptide chains. So polypeptide chains are our polymers. So in carbohydrates we had disaccharides, in fats we had triglycerides, phospholipids, but in proteins we call it a polypeptide. And polypeptides form the structural components of cells. They can have specific shapes, such as like enzymes. They are proteins, and you can find them embedded in cell walls as protein carriers for active transport and facilitated diffusion. So proteins are formed of these amino acid monomers, which contain carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. We've got plants who create their own amino acids, so they take in nitrogen, they fix it, and they can form their own amino acids. Animals can make some of their own in the liver. However, they have to ingest essential amino acids, which they cannot form themselves, so they've got to ingest them. You don't need to know which amino acids these are, but there are only 20 proteogenic amino acids, and that means 20 of them are found in proteins, but there are over 500, so the other 480 are not found in proteins. Each protein chain has an amino group, which is NH2. So I'm going to draw this on for you. This is an amino acid. So we've got on the right hand side we've got a carboxyl group, and on the left hand side we've got an amino group, and we've also got at the bottom an R group. So let's pause out there. So our amino group is NH2. On the left a nitrogen binded to two hydrogens, and on the right we've got a carboxyl group with a carbon double bond to an oxygen and a carbon to a hydroxyl group. Okay, so it forms a carboxyl group, COOH. Then we've got this R group. So it does not stand for a particular element. It is different in each amino acid. So for an example, glycine has a hydrogen. They vary in size, charge and polarity, some being hydrophobic, others being hydrophilic. Glycosidic bonds formed in condensation reactions. For lipids, we're looking at ester bonds formed in condensation reactions. In proteins, we call them peptide bonds, and once again, they are formed in condensation reactions. So let's have a look how this peptide bond is formed in proteins and how it differs. So when we join these two things together, just as usual, it's a condensation reaction, so we're producing water. So this one hydrogen and this hydroxyl group split off, the nitrogen and the carbon join together, and water is produced as a byproduct. Okay, so that is what the finished polypeptide looks like here. So we've got nitrogen, carbon, carbon, nitrogen, carbon, carbon. The carbon and the nitrogen here bind together. It keeps the double bonded oxygen. This hydrogen remains, but this hydroxyl group and the one hydrogen splits off, forming water as a byproduct. That is how we form our polypeptide chains. So polypeptide chains would be a great number of those amino acids. So now we've got four things to look at within proteins, and it's our primary structure, secondary structure, tertiary structure, and quaternary structure. So first of all, primary structure is the sequence of amino acids found in a molecule. Okay, found in a protein chain is the number and order of amino acids in a protein. Changing just one amino acid can alter the function of a protein. There are 20 to the power of 100 possible ways of ordering 100 amino acids. So the function of these proteins can greatly differ, even though we've only got 20 of them. Okay, so we can produce lots of different proteins, all with different structures and functions, as our primary structure can be altered. So our primary structure is basically just the order of amino acids, okay? It's pretty simple. Then we've got our secondary structure. So that's the initial twists in the shape of the protein, and they can form an alpha helix, which I've put in the diagram here, with hydrogen bonds between hydrogen and oxygen here, and you can also have a beta pleated sheet, which has the same hydrogen bonds between the oxygen and hydrogen, but also hydrogen bonds between R groups and hydrogen. But it depends on which amino acid it is. If the R groups are going to react like that, okay, that determines whether we're going to form alpha helix or beta pleated sheets, okay. Then we go to tertiary structure, so that's the further folding, and it forms this 3D shape. The coils and pleats themselves start to fold along the straight chains of amino acids, and it forms a tertiary structure. It's a very precise shape, which is held firmly in place by bonds between amino acids. We've got different bonds here, okay? So hydrogen bonds, which you know about, ionic bonds, bonds between carboxyl and amino groups that are part of the R group. So these ionize into NH3 plus and COO minus groups, which are strongly attracted to each other, okay? So I don't think you need to know much about ionic bonds. Just know that they do form between R groups, okay? So it depends on which R group the amino acid has, which determines sort of the tertiary structure. We've also got hydrophilic, hydrophobic interactions, and once again that is determined by this R group, and disulfide bridges and links. So there's only one amino acid which contains sulfur, I believe. If that's wrong, you can comment it. But that amino acid is called cysteine, and they contain sulfur within the R group, and if two cysteines bond together, there's a strong covalent bond between it called disulfide bridges or a disulfide link, and that is the strongest links in amino acids in the polypeptide. So if you've got lots of disulfide links, typically that protein will function better in high temperatures than other proteins. Okay, because when we get high temperatures, it breaks these bonds, and you'll come to understand that when you look at enzymes, which I've not made a video on yet. One more thing to look at with tertiary structure, we've got the 3D shape, which can be supercoiled and produce fibrous proteins, or spherical, which produces globular proteins. Okay, finally we've got our quaternary structure, which uses all these bonds again, but joins polypeptides. The tertiary structure, we've pretty much formed the one polypeptide, but the quaternary structure is the joining of multiple polypeptide chains, forming the 3D structure and the final protein, okay. So I've mentioned fibrous and globular, and here they are in a table. So let's compare fibrous and globular proteins. So fibrous proteins have repetitive sequences of amino acids, and they're usually insoluble in water, meaning they can form fibres which tend to have a structural function. So an example of this is collagen and elastin. We'll look at those in a minute. Okay, so globular proteins, on the other hand, coil up into a spherical shape, which we mentioned before. Any hydrophobic R groups are turned inwards to the centre of the molecule. Hydrophilic groups are on the outside, meaning the protein is soluble because more water molecules can cluster and bind to them. They often have very specific shapes which help them take up roles as enzymes, hormones, and hemoglobin. Okay, so globular proteins, spherical shape, hydrophobic towards the inside, hydrophilic towards the outside, and they adopt specific shapes. That is the outline of protein. So now we've just brushed the surface of fibrous and globular. We've compared the two. The main difference being insoluble and soluble for globular, structural function for fibrous proteins, whereas we've got a specific shape, so enzyme function for globular proteins. So let's have a look at some examples of different types of protein. So first of all, we've got collagen. So collagen is a fibrous protein. It lines artery walls to prevent them from bursting when withstanding high pressure. It's also found in tendons to connect muscles to bones, and bones themselves are made of collagen, but they're reinforced with calcium phosphate, which makes them hard so they don't break. So collagen, fibrous protein, structural function, it's just very strong and used to withstand high pressure in all sorts of areas within the body. Then we've got keratin, which is another fibrous protein. It has a lot of cysteine, which is our amino acid with sulfur in it. So we have a lot of disulfide bridges, so it's very strong. It's found in hair, nails. It's used for waterproofing, and it is impermeable to water. Okay, so helps prevent infection. Uppermost layer of our skin is formed of dead cells containing keratin, which provides an impermeable barrier to microbes and anything getting in. Okay, then we've got elastin. So elastin, once again, is a fibrous protein. It's got cross links and coils in its structure, making it strong and extensible. It's found in most living things where they need to stretch and adapt to their shape. Skin can stretch around bones and muscles, so skin's got elastin in it. It's found in lungs, allowing them to inflate and deflate. The same within bladder. So it also helps blood vessels to stretch. So elastin, like elastic, is used for stretching. Then we've got hemoglobin. It's a globular protein. It's got a 4 polypeptide quaternary structure, 2 alpha globin and 2 beta globin polypeptides. Heme groups with an iron in them for oxygen association, and we call that a conjugated protein as it has got these heme groups associated to it. Okay, a protein associated with this sort of group is a conjugated protein. It's globular, so it can dissolve in water, and when oxygen binds or something binds to hemoglobin, it turns from purple to red. Finally, well, not finally, we've got 2 more actually. Insulin, another globular protein. So we've got an A chain with an alpha helix and a B chain which has a beta pleated sheet. So you should know insulin from hormones. It's got disulfide links, hydrophilic R groups on the outside, making it soluble. Insulin is our chemical messenger. Another example of a globular protein. It is used to increase the uptake of glucose from the blood. Then we've got pepsin, which is an enzyme which digests protein in the stomach. In the protein, we've got 4 acidic R groups, which help the polypeptides stabilize in the stomach acid, and we've got lots of disulfide bridges, so there's cysteine amino acid helping pepsin to not denature within the stomach. Okay, so that was all we have on proteins. Now a very short bit on inorganic ions. It's not one you can really teach, it's just one you've got to learn. There's nothing more about it. So inorganic ions are an ion with an electric charge. You can get cations or cations. I know no one calls it cations. With a positive, so a plus ion, and anion, which are negative ions. Right, so this is a picture I took from my textbook, and if you've studied the whole course, you will pretty much recognise all of these ions at some point in the course. They all really come up again if you notice it, okay? And we've got in blue here are cations, and in green are anions. And I don't want to spend too long on this because it's not really a matter of not being able to understand it, it's a matter of you've just got to learn this table. Okay, so we've got calcium Ca2+, nerve impulses, acts as a cofactor, bone formation, You notice when we make the muscles contract, that's when we see calcium come in. We've got sodium, nerve impulses. You should know all about sodium influxes for action potentials. It's used for muscle contraction and regulating fluid balance. Potassium, it's K+. Once again, nerve impulses again. Remember that period of hyperpolarization, we're pumping potassium back into the nerve. It's used in muscle contraction as well. Activates essential enzymes needed for photosynthesis. Hydrogen H+, if you do not know this element, you should not probably be doing biology. It's all over, it covers lots of topics. Respiration, photosynthesis, when we're looking at oxidative phosphorylation and photophosphorylation. Affects the pH of substances, so the more hydrogen we've got a lower pH, so we get an acidic substance. Ammonium NH4+, one of the harder ones to remember. It is absorbed in the soil by all plants as an important source of nitrogen, so it's found in the nitrogen cycle. If you look at the nitrogen cycle, you'll probably remember ammonium's function. Then we've got nitrate, NO3-. So these, now we're looking at our anions. It's once again in the nitrogen cycle. It's an important source of nitrogen, used to make amino acids, nucleic acids, etc. Another anion is our hydrogen carbonate, used as a buffer which helps maintain pH of the blood. And we've got chloride, so the chloride shift, very closely linked to hydrogen carbonate. We see that in the same sort of thing. It is also involved in some nerve impulses and acts as a cofactor for the enzyme amylase. And that question has actually come up a few times, and I always get it wrong because you never really look at small details like that, but it is important to remember all these sort of details. So chloride is a cofactor for amylase enzyme. Phosphate is PO4 3-. A harder one to learn, just learn it. We see it in photosynthesis, respiration. It's found in nucleotides. We've got that phosphate group, ATP, triphosphate, ADP, diphosphate, AMP, monophosphate. You see it all over the place. Phospholipids, calcium phosphate to strengthen bones, which we just spoke about. Finally, we've got hydroxides, which is OH-, and it's another thing that you see a lot, affecting the pH of substances, makes it more alkali. Okay, so the more hydroxide ions, the more alkali the substance, the higher the pH. So that is all on proteins and inorganic ions. Next video, we're going to be looking at biochemical tests, colorimetry, and chromatography.